Hello everyone, my name is Daniel Cazares and I am a part of the Plants in Space group at SEAS, and this is our presentation, Cultivating a Galactic Garden.
Hello, my name is Ashley Kroeger and I'm from Arlington, Virginia.
Hi, my name's Liz and I'm from Rogers, Arkansas.
Hi, my name is Isaac Hildebrand and I'm from Salt Lake City, Utah.
Hi, my name is Daniel Cazares and I am from Chaparral, New Mexico.
Hi, my name is Rebekah Humphries and I'm from Tucson, Arizona.
Hi, my name is Rowan Hankins and I'm from Salt Lake City, Utah.
The presentation is split up into four main sections: the background, the Hitchhiker Seed Project, the ZQ project, and the conclusion.
Plants play a crucial role in the future of space missions, both as a sustainable food source for astronauts and as an important resource if humanity ever hopes to establish life on the moon and, eventually, Mars. Changes in gravity, both brief and prolonged, place an immense amount of stress on plants. For example, stress can cause disturbances in the calcium ion distribution in plants, which is important for signaling, membrane stability, and maintaining cell wall structure. This distribution weakens the cell walls and overall structure of the plant, making them more susceptible to damage, like stunted plant growth. In the absence of gravity, another stress adaptation from plants is the loss of direction perception, resulting in erratic growth patterns. Additionally, lunar regolith, or lunar soil, is highly hydrophobic in nature. This nature negatively impacts the plant's absorption of water and nutrients, further compromising its health and development. To better understand the effects of microgravity on plants, radish seeds that were previously flown on a parabolic plane were planted in 6 different sample containers, each consisting of 9 seeds. Half of the sample Petri containers were flipped upside down twice a day, once in the morning and once in the evening. Their irregular growths demonstrated the abnormalities caused by exposure to varying gravity. The results showed that the spun seeds germinated at the same rate, but the roots formed a loop around the center of the seed.
The composition of lunar soil is drastically different from Earth soil because of the moon's airless body that doesn't have the benefit of geological processes of water and wind erosion. Its high composition of mineral and lunar rock fragments, as you can see in the image, presents the problem of hydrophobic properties. It is compact with fine grains of dust particles that are sharp with fresh fractured surfaces. Water movement in lunar soil has poor water retention and rapid drainage because of the regolith substrate, high porosity, and fine particle composition. The fine particles in lunar regolith substrate disrupt capillary action, preventing even water distribution throughout the soil. This means that water either drains away quickly or evaporates, making it difficult for roots to access the moisture they need to thrive. Furthermore, there are no natural components to help retain water and nutrients. Understanding lunar regolith substrate water distribution is key to the future of a crop growth because it will develop effective irrigation and soil management techniques. Based off of the ISS Veggie and Advanced Plant Habitat Systems, exploring long-term effects of microgravity and identifying patterns of plant growth will help analyze seed germination failure. Studying plant patterns in space leads to new insights into their biology and stress responses. Our mission is to pioneer sustainable agriculture in lunar regolith by drawing from ISS methods, and we aim to address the unique challenges of water movement as well as nutrient retention in lunar soil.
The Hitchhiker Seed Projects. We will send Arabidopsis seeds and seedling samples up in a parabolic flight. These little seeds hitch a ride with other experimental payloads on the aircraft and thus are referred to as hitchhikers. So, what will we be flying? We will fly 2 samples, seeds and seedlings. Arabidopsis seeds will be placed in an airtight pouch. The seedlings will be placed in water agar, which will grow 3 days prior to the flight. There will also be seeds from the same batch kept on the ground and grown at the same time and under the same conditions, other than the microgravity. The seeds kept on the ground will be used as a control group to isolate exposure effects. Effects of microgravity on Arabidopsis growth. Once the seeds get back from their flight, they will be grown and studied for long-term effects. The factors that will be observed include root growth, plant health, and how temperature can affect growth of the exposed seeds. Utilizing the translucent of the agar, the pattern and length of Arabidopsis roots will be observed. Previous NASA studies have recorded erratic root growth and direction. Pictures will be taken of root patterns and compared to a color chart and a root chart to monitor the plant's calcium levels and effective root development. Root development is a critical phenomenon in growing plants in varying gravity because it is necessary for proper growth. The reason plant health is a huge topic to look into is due to the fact that plants experience stress in microgravity, which usually leads to abnormal or stunted growth. Leaf size is also essential for growing plants on the moon because without large, good quality leaves, plants are inedible. The seeds will be planted in small pots and moved in environments with different temperatures. The first group will be placed under a heat lamp at 40 degrees Celsius. The second trial will be placed in a fridge at 4.4 degrees Celsius. The third and final trial will be kept in a temperature-regulated room at 22.5 degrees Celsius. The purpose? To observe extreme temperature tolerance of exposed seeds. This is very important because the moon has extreme climate conditions, with temperatures going all the way up to 127 degrees Celsius and getting as low as negative 250. The different environment will help us understand how exposed seeds compare to regular seeds in varying climates. To monitor seed temperature tolerance accurately, there will also be a control group of seeds that have not been exposed to varying gravity, under the same conditions in order to isolate the effects of microgravity on the exposed seeds. During the flight and before starting the growth cycles, the seeds will also be kept in a temperature-controlled room with conditions of 22.5 degrees Celsius.
Our next slide will be about logging our growth. In order to maintain the growth of our seed samples, we will each keep each other accountable by keeping a data and watering log. In addition, there will be 3 repetitions of growth cycles to make sure the data provided is accurate. Each of the 3 repetitions will be flown and exposed to microgravity in a parabolic flight and will each be placed in different agar samples. And we will also be using Arabidopsis. This was because a huge benefit is that Arabidopsis has a lot of studies that we can compare our data that we find to. We will also have a separate control group to monitor, that way we can make sure we record and log all changes done when exposed to the different factors, like the temperature. We will also be recording and logging data in our schools for the most part. This is because we want to educate our local communities in our respective areas in order to educate about the importance of understanding microgravity and its effects.
Next up, we are going to talk about our experiment with water diffusion through regolith using the Z-Cube. One issue that will be a problem when it comes to growing plants on the moon using lunar regolith is the fact that, as was said earlier, lunar regolith is very fine and very compact. This makes the lunar regolith very hydrophobic, and it makes it very difficult for capillary action to occur, which means that water cannot diffuse through the lunar regolith. It is important that this issue is overcome, as plants grown in lunar colonies will have to be grown in lunar regolith, considering that it is very abundant on the moon. There are several different types of lunar regolith depending on what region of the moon you are on, but for this experiment we used a Highlands lunar regolith simulant from NASA Astro Materials. To help us conduct this experiment, we are using the Z-Cube, which is a prototype microgravity sensor developed by Twig Space Labs. As seen in the graph on the left, the Z-Cube contains an LED light at the top, a spot for an experimental payload, a sensor stack, and that sensor stack includes a camera. The sensor stack also includes an accelerometer, humidity and pressure sensors, temperature, and a magnetometer. And we will be putting a payload into the Z-Cube for a parabolic flight. Okay, so the goal for the water diffusion through regolith project is to learn more about potential water diffusion techniques for lunar regolith. The primary measurement that we will be taking is the moisture level of the regolith simulant. The variables in this project independent will be the microgravity environment, as well as the hypergravity and regular gravity, as well as everything in between during the parabolic flight, as well as whether or not perlite is useful in assisting water diffusion through lunar regolith. The dependent variable in this project will be how well the water is absorbed and diffuses through the regolith simulant.
Pre-work. Before reaching our final product, many experimental procedures were held to discover the materials needed and the methods to perform these by. With the goal of this project in mind, we utilized a wick, perlite, and Orbeez.
After a successful drop test where we each experienced microgravity, these were our findings. The wick spreads water efficiently, but not throughout the entire sample of the regolith substrate. Perlite took an extended period of time, but proved to be more thorough in means of range. Finally, the Orbeez absorbed the water before reaching the regolith. The purpose of the additives are to space out the fine particles within the substrate. This will create a less dense environment for the water to flow through, resulting in more efficient capillary action. We decided on perlite as the most effective additive for this specific experiment. Design development. In the experimental stages of this project, we started out with a simple design featuring a tube filled with lunar regolith substrate and tilted at an angle for optimal video angle capture from the ZQ. The water would be added right on top before takeoff. For our second design, we added wicking and perlite to the lunar regolith substrate, testing the effectiveness of additives. For our next design, we took out the wick, figuring that there were too many variables involved with the testing. The issues experienced in the first couple of designs mainly revolved around the water mixing with the regolith substrate samples before ever experiencing the microgravity. Final design. These models represent the experimental structure of our final project. We first separate the container with a tube, and then create a secure barrier with hot glue. On each side, we sampled lunar regolith substrate. To one of those sides, we added perlite. Right before the beginning of the experiment, water will be added to the tube in the sensor. The idea is that during the microgravity, the water will begin to crawl up the sides of the tube and out onto the surrounding samples. After the variations of gravity have been experienced, we will observe the movement of the water using the camera on the ZQ. We will also use the needed measurements on the sensor stack, including humidity.
This slide contains the procedures that will be carried out during the parabolic flight and in the hangar beforehand. The first step will be placing enough lunar regolith simulant on the left side of the barrier and well to cover the bottom of the lid. After that, equal parts lunar regolith, simulant, and perlite will be mixed together. And for step 3, that mixture from step 2 will be placed on the right side of the buried well enough to cover the bottom. Next up, the center well will be filled 3/4 of the way with dyed water, and the water is dyed so that it's easier to see when the water leaves the well from the camera. Finally, the experimental payload will be placed upside down in the ZQ2 stack, and the light and camera will be started. Our data analysis of this project will be checking the camera footage and looking for darkening of the regolith simulant, which will indicate the diffusion of water through it.
Up next is our conclusion. For our conclusion, we will quickly skim over and summarize both of our project data. Up next is the conclusion, the Arabidopsis. The Arabidopsis growth and development project seedlings will be sent into a microgravity parabolic flight, as previously mentioned. The Arabidopsis seeds will then be distributed to each team member's schools with the respective experiments conducted in a classroom setting in different temperatures, and the growth and data will be recorded afterwards. Then, monthly meetings will be held between team members to discuss all of our findings, and our goal is to better understand the effects of microgravity on plants. Data will be shared between classrooms, resulting in a national collaborative experience.
Utilizing the Z-Cube's tools, we will monitor our payload, which will hold lunar regolith simulants, one with perlite and the other without. They will be placed in a microgravity environment in order to further understand how water and regolith interact in a microgravity setting. We would like to extend a special thank you to the following people. Laura Tomlin, our wonderful project mentor who aided in our research and agreed to carry our hitchhiking seedlings on the parabolic flight. Margaret Baggio, the SEAS program director. Selena Miller, the SEAS program manager. Jacob Torres, who provided an immense amount of information and took the time to answer our many questions. Dr. Mickey Sodi, who met with us virtually and contributed to our initial research. Becky Fritchie, the best van driver we could ever ask for. And all of the other SEAS program members who we couldn't have done this without. Thank you for your time today, and we will now be taking any questions that you may wish to ask.